Zuckner, who is a professor of neurology and human genomics at the University of Miami. And Stefan typically tells us about all the exciting advances in human genetics that has enabled us to identify many of the causes of CMT. But a particular type of CMT that we're very excited about is CMT SORD. And this is sort of a tour de force of what Stefan has done over the years in CMT, because it required extensive next-generation sequence analysis to identify the gene. He was able to develop models of this, and it's led to clinical trials. And basically, Stefan has been behind all of these stages. So we're going to ask him to tell us about SORD neuropathy from lab to treatment. Stefan?
Thanks, Mike, for the invitation. As you can tell, very many difficult personalities involved here in this field. Do we have slides? Oh, you're supposed to.
You are Zuckner.
TBD.
Yes, to be determined, your title.
So, perfect. Got it. Awesome. So, as Mike said, I'm very happy to summarize where we are with this very important subtype of CMT, CMT SORD. And as Mike said correctly, it reminds me a bit of what I imagine you get taught in business school, these case studies. You know, you learn something from case studies. And I think this is almost a perfect case study. It, as I said here, the story has it all. So I hope you enjoy that from this perspective. Here are my disclosures. So first of all, the baseline where we are today is that still, numbers vary, but it's safe to say that 50% of CMT2 patients do not get a genetic diagnosis today, despite efforts, or even 70%. So, and the question has been for geneticists, where is the missing heritability? How can we close the diagnostic gap? Could it be a large number of rare disease genes, maybe dozens more, that make up this difference? Is it something that is really hard to study, like digenic or multigenic inheritance? Or has it to do with limitations of technology, and maybe there is much more to discover in the non-coding genomic space, for instance? So this is where we started out 5 years ago, when we were about to discover this gene SORD. And I had a fellow in my lab at the time, Andrea Cortese from Italy, who was actually working at UCL in London. So the idea was, the basic hypothesis or question was, could there be DNA variants that are not seen in controls, but are seen multiple times in patients with CMT? That sounds easy enough in a way. So to answer such a question, first of all, and this is so where the puzzle pieces come together to be successful in this field, is you have to, try to accumulate high-quality data, aggregate data. That is clinical data, and is where the INC consortium is invaluable, but also find ways to aggregate genetic data, but not just sort of putting them on a hard drive, but have them available so you can analyze them, and not just one person can analyze them, but make them available to a lot of collaborators. And this is what we have done for over a decade now with the Genesis database, which is also supported, for instance, by the MDA and other patient-driven organizations. So this is a screenshot, basically, of this database where you can query over nearly 3,000 CMT data sets now. And Andrea tried different scenarios, and he sort of came up with this here. And as you can see in this red box, all these hits, what they have, they all fall in this gene SORD. They all have the same mutation, and the code 166 stands for CMT. So there's clearly an enrichment of CMT cases there. Here, this is an ALS case. And they all, as I said, they all share the same variant. In fact, there are so many of those that what really happened here is that we had dismissed this prior because that is too common. This is probably an error. But Andrea was persistent, and so it was good. So first of all, what is SORD? SORD encodes a protein, an enzyme, sorbitol dehydrogenase. And when you look this up, you immediately find a pretty large literature on this, mainly because sorbitol dehydrogenase or sorbitol is been involved or has been involved in diabetic neuropathy for decades. It's sort of a textbook pathway for diabetic neuropathy that medical students learn. Even drugs have been developed to try to influence this pathway with sort of mixed success, I would say. So that seemed like a very good candidate, except that we were still troubled by the high frequency, a high number of families or patients. What also is kind of striking, we didn't have a single larger family that could do linkage analysis or really extended segregation studies. The signal came from many sporadic cases. It's a recessive disease, biallelic, so this kind of makes sense. But it also shows you the power of bringing a lot of data together, because then you can use these sporadic cases. You develop the power just by the numbers. So we started screening more patients, of course, that were not in our database. We found additional mutations that are listed here. But what stood out and is unusual in CMT for sure is that one allele, the one we saw first, is really the most common allele by far. In fact, even today, there are very few patients where this allele is not involved, at least on one chromosome. So, okay. When we looked up this particular variant in NOMAD, which is a large database for controls that do not have CMT or any disease we are aware of, we see that the frequency, the occurrence of this is actually quite high. In fact, 1 in 200 people in the population are carriers. They carry one copy of this particular frameshift mutation, 1 in 200. That's pretty high. To put this also into context, here are some of the most common alleles in the CMT field, alleles that are recurrent and that have been seen in patients over and over again. And you see that the SORD variant we're talking about here is by an order of magnitude more common than the most commonest known, except, of course, the PMP22 change. And from this, we can calculate a homozygosity frequency in a population, assuming sort of normal distribution, of at least 3,000 patients that are homozygous for this particular change in the United States. So, and that pretty much makes it the most common recessive CMT just by itself. And we think it explains about 10% of all the axonal recessive CMTs and even DHMNs. So one question we had early on is how come we only now in 2019 it was see this signal when we have studied these cases for 2 decades or so. And the answer probably lies here. SORD is a gene that has a pseudogene, a highly similar, highly homologous gene copy that is not active, we call pseudogene, just upstream of it. And the name for this is SORD2P. So SORD2P is a completely inactive transcript but it is highly similar, the sequence still to this. And what happened here is when you do exome or genome sequencing, the bioinformatics pipelines that realign these short reads back to the genome, they have trouble to decide if a read should be aligned here or down here. And here is the zoom in right where this most common variant is. And this is the difference. A C is missing here, or is added rather in the real sort gene. And you see the red letters here show the difference between the two loci, and you see they're highly similar. So at the time, 2019, we looked at this. For instance, there was no NOMAD. The prior database was called AXEC. And AXEC had the allele frequency for this variant completely wrong. In fact, we went back, we developed a nested PCR to really determine this by hand, so to say. And the reason was the whole world was using an alignment software that had trouble dealing with this locus. So the new GATK version 4 and later is quite good at differentiating between sort and sortropy. We, at the time, used a different pipeline. We used a different software, and that's probably one reason why we stumbled across this first. So it shows another aspect of this. I mentioned data aggregation, but also you need great skill and talent around you to do bioinformatics analysis and also sort of be willing to think unconventional. So, okay, we were a bit more convinced now SORT is a good candidate. We were able to acquire fibroblasts from patients. And so here are control fibroblasts, and these are from SORT patients. This is a Western blot, and these are carriers. And you see it's a complete loss of protein. So it's a homozygous truncation, leads to a complete loss of protein, as often is the case in these diseases. So up here we see the pathway, which is well understood, the polyol pathway. It starts with glucose and aldose reductase, turns this into sorbitol, and then which is considered toxic, and is really hardly present in normal tissues and blood. And then sorbitol dehydrogenase, or SORD, turns this into fructose. So often the polyol pathway is described as a pathway when you have high glucose. It's sort of a release mechanism from the high glucose to turn it into fructose. Now, if you don't have this anymore, then you accumulate sorbitol. And that's exactly what we find in patients. When we just measure this in blood, but you also can measure it in urine, which is now has been developed by the Mayo Clinic, you see many-fold increase of sorbitol in these cases that don't have any sorbitol dehydrogenase anymore. And that is a fantastic biomarker right there. Okay, so it's great confirmation. It's a fantastic biomarker. So that when we saw this, we were super happy, and we reached out to a friend and colleague who does fly genetics, Grace Tsai, in Miami, and she developed with us a fly model that does not have SORD. In fly, it's actually called SODH2, and these flies, they have defects in their eyes, and they actually develop. When you look closer here, these sort of holes, the neurons in the fly eye, they develop these vacuoles. And when you quantify them, so this is 2 days old fly, 10-day old fly. In the knockouts, you see the vacuole number increases and also the size increases over time. You can actually measure in these flies how well they can climb up a glass wall, and as they age relatively late, actually, in a fly life anyway, the knockouts, are getting worse than the wild types with age climbing up a glass surface. So in a way, we were able to show in this fly model that there are some real physiological changes and also behavioral changes when you knock out SORD. So then the question quickly came up because of the whole diabetic neuropathy history. Well, is this potentially a treatable disease? And remember, CMT has today no real treatment. So again, the sorbitol pathway, we don't have this anymore. Sorbitol goes up. How about we block the aldose reductase enzyme? Then glucose would not enter the pathway and sorbitol would not accumulate. So that's the idea behind these drugs called aldose reductase inhibitors, or ARIs. So fortunately, there are several of these. Several have gone through clinical trials. The safety profile, I say good safety profile, it's actually somewhat questionable. It's not perfectly good, which has led to these drugs never being really approved in the United States or in Europe, but they are available in some Asian countries. Also, the efficacy in diabetic neuropathy was never quite clear in the US studies. But they are available for research purposes, so we tried them on the fly model. And as you can see, here are the two drugs. Here is a wild type and a control fly. These are the knockout, these are actually fibroblasts. But you can start fibroblasts, and these are Drosophila brains, same effect. When you treat them with the drug, the high sorbitol levels come down to near normal levels. Okay, so the biomarker sorbitol is highly effective to measure that you can influence this pathway. And then again, flies here treated. We're measuring the ability to walk up a glass surface. And again, you see the red here, how the older flies decline faster in their ability to walk up, while the treated ones, they are rescued, definitely significantly above the degradation we see in the knockout flies. So these are great results, and in fact, there is a very new, fairly new ARI on the market, or not on the market yet, but it's called govorestat, or AT007, and David already mentioned it. It is studied by a company called Applied Therapeutics, and they have approached us, and they funded Gray's Zeiss lab to try the same experiments with their ARI, and sure enough, they see the same effect. So also this new ARI, in fact, can rescue the fly phenotype. So this new ARI actually can penetrate the blood-brain barrier. That is a major advance. And as we already heard, within 2 years from publication, the company organized a clinical phase 2/3 trial. So that was pretty much filled after only 2 years of publication. We just passed the 1-year mark of this trial. And it is just unbelievable that this can happen so fast. And again, another key point here is the INC and friends. They had prepared for this for over a decade, 15 years. They have accumulated patients in their clinical databases that were readily available when we needed them for these types of studies. So the trial could fill very fast with patients who were motivated and had good clinical data. And I'm not personally involved in this trial. I have advised in the beginning on the design, but this is also just basically a screenshot from the website of the company. And as you can see here, just a couple of weeks ago, they released the first results after a 12-month readout. And as David mentioned, there are several points that trend in the right direction. For instance, there's a sustained reduction in the sorbitol levels. That is what you want. And it seems like there is even some objectively measurable effect on the CMT form and on the CMT high. So it's too early to be really excited, but this is not bad at all. So in addition, we use this rich data set from the INC to start a cross-sectional natural history study. And we also reached out to other colleagues beyond the INC, and we identified about 150 patients with SORD that had also clinical data available. And some of the clinical data was like 10 years old, and they had even been seen multiple times. So that's a very nice study that will soon be submitted for review. And these are some of the details, quite typical, I would say. It's mostly a distal length-dependent neuropathy. And again, another puzzle piece here, natural history studies are so important to support the clinical trials, support their interpretation and readout. So another piece is to make this complete is we always talk about, okay, now we need to model it also in vertebrate and mammalian models. There has been attempts to create mouse models for SORD, mainly because of the diabetic neuropathy link, but they have sort of failed to replicate the phenotype of a neuropathy. So we decided to create a rat model, and great thank you to the Hereditary Neuropathy Foundation. They supported this effort. So we have the rat model now. It's studied. In fact, these data will appear in Brain in the next month or so. First of all, sorbitol levels are, as you can easily see, elevated in serum, peripheral blood. In the CSF, even more so. That is something not really been studied yet in humans. We have some early human data. As you can imagine, it's hard to get this. But it seems like sorbitol is also produced in the CNS separately, and it has trouble going through the blood-brain barrier. This could be important for pathophysiology, but it's also in other tissues. So it seems like sorbitol is high everywhere. Is it a timing issue?
You're okay.
I'm okay, good, because he was waving. So wonderful what you can do with a rat model. Now you can study all the tissues you want. We also looked at other biomarkers such as neurofilament light. It's a unspecific but well understood biomarker. And clearly there is we can measure it here. So now we have two biomarkers potentially that we can also look in humans for. The behavioral studies very much mimicked what we have in humans. This is not a very severe CMT. These animals have measurable differences starting at 6 months of age, roughly. This is data from 11 months and 13 months. And these are different measures of behavior. We also measured nerves with nerve conduction, velocity, and their differences. So all mimicking sort of the human findings. Now, what we have in addition now, which we don't have in humans easily, is, for instance, nerve biopsies. So we started studying these nerves extensively, and this was done by Steve Scherer, and very exciting to report that these nerves clearly have defects. As expected, they have somewhat thinly myelinated fibers, some onion bulbs, but also very much signs of axonal degeneration. What was most striking is these large structures here. When you actually get closer, these are actually nerve fibers. So they have been sometimes called balloon fibers and have been seen in experimental mouse models 20, 30 years ago of diabetic neuropathy. So these are very, very unique structures that we're just starting to study deeper. And they could be somewhat pathognomonic for this condition, actually. So very exciting to have these models available to do deeper studies. So with this, I hope I convinced you, this study has really a lot of great pieces in it that we always try to strive for. I'm going to go through some of them here. So gene identification, obviously, it's important to find these genes. It's great to find something that's more common. So, several thousand patients now get a diagnosis. And it also pinpoints the limits of short-read genome sequencing. Because of the homology region, long-range genome sequencing will be much more effective here, and for similar loci, potentially. So this will help closing the diagnostic gap, for sure. Biomarkers, we always want biomarkers. They're very important. Here's a disease CMT type-specific biomarker, which is also reactive in real time when you treat. So this is super valuable. Animal models, we already have a valuable fly model. We have a brand new rat model, which I think is going to be fantastic to finally understand what sorbitol does to peripheral nerves. This has been very fast to go go into people as a therapy, and that would be, if it works and it continues to look good, would potentially be the very first treatment for CMT and be a real landmark for the field, and kind of demonstrating that it can be done. And then this point, I always think very fascinating. Rare disease can inform common disease. Sometimes rare disease is like, this is the rare disease, but how our work can also very much inform more common ailments. And in this case, there is clearly a connection here between CMT SORD and diabetic neuropathy. And again, the rat model probably will help with some of that, clarifying some of this. And I just really want to acknowledge again, the Inherited Neuropathy Consortium has been a tremendous resource, has been funded now for 15 years by the NIH, and has created an amazing group of investigators and also resources that we hope will continue. And these two individuals really had their hands on a lot with all of these steps here. Adriana Rebello is, we call it lab mom. She's the senior scientist in my own lab in Miami. And Andrea, he's back in London now, but he's still very active when it comes to SORD studies. Thank you very much. Oh, good. Any questions? We have time. Yeah, so can you use the microphone? That way it's hard for people to hear the question. I think, yeah, go ahead.
The question was if there's any consideration for modified Atkins or low glycemic diet similar to treatment for Duza syndrome for CMT SORD.
Yeah, excellent question. We ask this all the time. Can there be dietary treatments? Also, another question would be, can I lower my sorbitol intake through food and that sort of thing? We have actually tried with these rats to put them on a diet, low glucose. It didn't make a difference. Sorbitol itself, it's so high in these patients that you couldn't even dietary get there, I think. It's so, we don't know, there's no hard data, really, but probably not. That's my current judgment.
Great presentation, but that was my same question. Ketogenic diet.
Are there any active plans to study pediatric populations with these treatment options?
If you looked into pediatric populations?
No, to treat pediatric populations.
Is
there anything in play?
So, okay, the current trial inclusion criteria, I'm not sure what the age, but do you know, Mike, when they?
16 or 18, I forget which.
16 or 18 and up. So first of all, the mean age of onset is 18, I believe. plus minus 8 years or so. So it's not a super early type of CMT, but children can be affected for sure. I hope the way it will go down might be this current trial. Let's hope it's successful, and the drug gets marketed. I would imagine the company would then shift focus to broaden the age range where it can be prescribed to. That's my current, how this probably will happen.
Can you use the microphone, please?
Does the sequence homology impact the ability to detect this with commercial genetic testing?
That's a great question. It impacts it for sure. In fact, not all the major clinical commercial labs offer this test today, even almost 4 years later, for that reason. But it is offered by a couple labs, but it causes issues. Yeah. And there is even more. We just recently, that's not published yet, Andrea found a change which is a large inversion, like a very large inversion, that you can absolutely not see with any regular test. You have to do long-range sequencing or a very specialized PCR. And we have started screening patients, and we found that 10% of the cases with SORD have this inversion. So frankly, the best test right now, which is not available, is a sorbitol test, like a biochemical test. As some of you certainly know, a Mayo Clinic is working on that, and I think it's getting close to be released. You can probably get it today when you contact them. In Miami, we offer a sort of research-based blood serum test. If you have cases, we're happy to do this on a research basis. So the tools slowly getting there, and I would think the trial outcome would expedite the need for these tools.
Can I ask you a question?
Yeah, Allison.
For Allison Moore, H&F. For the sorbitol test at Mayo, Dr. Shine mentioned it to me yesterday. Is there a charge for that, or can patients do it for free?
I don't know, Allison.
Okay.
I have to.
If we could,
Not sure.
I can answer that.
Yeah, sure.
So there is a charge, and I don't want to say the number too precisely because I'm not sure of it, but I think it's in the hundreds, and it is possible in some cases that we've had insurance cover it, but it's not guaranteed.
okay, thank you.
Okay. So can you exit, Stefan? Or I can do that, thanks. Yeah. Lovely talk, thanks.